And a key thing, though, regardless of what they ask, is to think about the statistical implications of that. So certainly, either if it was planned from the start or something happens where they add this as a modification, once you start looking at efficacy, that's inflating your type 1 error if you don't adjust the statistical analysis for those added looks, essentially requiring tighter evidence each time. More evidence early on, and certainly still more evidence at the end because you've looked multiple times. And there are many methods to do that, and so it's important that you agree, certainly as soon as you realize you're going to need that plan, on what plan would be used to make those decisions. What stopping boundaries would be consulted, right, in terms of essentially there's a continuous boundary in terms of amount of information collected so far, test statistic at that time, and whether or not you're above that level or below that level would be the reason to stop. And just to convince you, or just perhaps to make it more intuitive, as to how quickly additional looks can inflate your false positive rate. So that false positive, the idea that there'd be two arms where there's no difference at all. But if you keep looking, maybe you have more chances that just random streak could create a difference. And so on the first column here, we have the number of looks. And say 1 would be a trial with no monitoring. At the end, you just look and you do a test at 0.05 level. At the extreme, you do 10 looks. If you did 10 looks, say equally spaced, and you always use 0.05 to decide at each of those 10 looks whether or not you would stop in terms of the difference, the p-value was less than 0.05, at any of those 10 looks you'd stop, you can see that actually your error rate would be 19, almost 20% instead of 0.05. If you did 5 looks, your error rate would be almost 15%. So it does increase quite quickly. So even if you're doing 2 looks, I think you should adjust. And that's just if you're doing it equally spaced. There's some ways you could game the system in terms of doing the looks when trends appear. This is a little bit mathematical, so I'm not going to explain exactly how I define this worst-case spacing. Let's just say there are ways to game the system and the timing of the looks so that the type 1 error rate could be much larger, as high as 45%. And it sounds ridiculous, but I've certainly been involved in, not in trials, but in analyses, say, with investigators, and these are mice, which are a little bit easier to recruit, say, to your trial. That you do an analysis and you realize, the p-value is 0.06. Let's add another mouse and not quite there yet. Let's add 5 more mice. Hey, now it looks kind of convincing. Let's publish. Right? There's something inherently wrong with that. Let's add mice until we get a difference. You need to adjust the 0.05 level, and so that is an example of worst case, or getting close to worst case. That's not equally spaced at all. Okay, so once you set up these boundaries, which are going to be, okay, I'm not going to use the 1.96. I'm going to make it harder at the beginning. Maybe I'm going to need a 3. There's some mathematical principle that's going to guide us. The thing that's interesting is that if you read examples of these trials, you can kind of see what random streaks do. But there's all kinds of examples where you get incredibly close to the boundary and you're all ready to stop, and in the end, a completely opposite decision is made, which emphasizes, I think, just the need to have the structure, because I think randomness is a little counterintuitive. Like, we think equally spaced is somehow random when actually that's quite regular and hard to achieve. So the Coronary Drug Project was one of these large cardiovascular trials. And once they set up the boundaries, they actually reached minus 1.96 3 times, which isn't reason to stop because they needed stricter rules because they were looking multiple times. And in the end, they wound up at 0, no difference. There was another trial in the literature that's historical. It was called MUST. And this was looking at whether or not if you did guided treatment of arrhythmias with this electrophysical testing, whether that would be a benefit or not to the usual unguided treatment. And that appeared harmful right away and stayed that way for quite a bit, and then in the end actually proved to be beneficial. So I think it's these sorts of examples emphasize the need of a sort of interdisciplinary body that can really think about the pluses and minuses or the risk-benefit to the patient, and also, if you stop too early, you're not going to convince anyone that the treatment was harmful or beneficial. That's the worst-case scenario. that you have to start all over again. And there are examples of that, where in the end, people were convinced neither way and were still interested in the treatment and they had to start all over. And to give you an idea of how these boundaries work, and actually that there is something to choose, although I think there's one common one that people typically do. But so you think about this is the Z-score boundary. If we did one test at the end, we'd be down at 1.96. If you do lots of tests, think about here on the x-axis, we would have an interim analysis. Maybe we do 4 looks. So you can think this is in time our looks. And you want to make it, the O'Brien-Fleming principles, you want to make it really hard to stop early on because you want to be swayed by those streaks in the data unless they're really convincing. And then that way you also benefit by having this sort of steepest descent. You wind up with the smallest possible penalty at the end where it's not 1.96, but it's just a little bit above 2. So you have some chance of stopping early, but maybe not a ton. Hey Biddle Pedo and the Cat in the Moon. They say we're going to do this really simple. P less than 0.001. If you're less than that at look 1 or 2 or 3, you stop. That's actually 3 times that is 0.003. That's barely an inflation if you think about it. I could make my p-value at the end 0.047. And so I do 3 sort of even amounts of evidence across these different looks, and then at the end I only have a tiny adjustment again, which is thought is desirable because the bigger the adjustment, the more people you need for that convincing trial at the end. And I say that there's a lot to be said for simplicity. If there's not a lot of staff, not a lot of experience in the statistical team, everyone can implement this boundary. The negative is, basically make it hard, and then kind of relatively not so hard at the end, is that you could have had evidence here that was towards a benefit, but not quite at that 0.001 level. And then you could have had all of the data accumulating that was in the wrong direction and still wind up less than 0.05, somewhere between 0.00, say 2, and 0.05. And there's something logically inconsistent about that. That you wouldn't have stopped for benefit at a p-value of 0.002. Then you had a bunch of a negative trend accrue and then stop for benefit. And so mathematicians sort of took a hold of this and tried to find a compromise that would achieve these goals in a way that would be logically consistent so that you couldn't somehow get stronger evidence by reverse trend. Pocock wanted it to be easy to stop early on, concerned about safety, but that boundary has similar problems with the pedo and is actually quite an expensive penalty in terms of needing a lot more people in the end, and I think he actually recommended against his own boundary. It seems strange to have the same, well, we'll leave it at that. The overwhelming, I think, particularly for large phase 3 studies, the overwhelming way that people do it is they find this thing they call alpha spending, where you consistently sort of spend some of your alpha, your 0.05, that's your allowable false positive rate, across these looks that for this boundary, magically, you don't even need to say how many looks or when the looks are happening. And you can have different mathematical functions and achieve this spending function, which basically spends the alpha, think about it in the steepest way, the fastest way, and still wind up spending 0.05 at the end of the trial, or spending it the slowest in terms of it spending very little alpha. We've got sort of 0 to 0.05 on the y-axis here, and then spending it all at the end towards the final analysis. That's the O'Brien-Fleming spirit, but done in a way that we don't have to specify the number of looks, but instead always just adjust for the percent of data in versus the percent of data that's left. And these two statisticians, Gordon Lan and Dave Demetz, came up with this clever way of doing the monitoring boundaries. And so it's become a pretty standard way to do trials. You still see the hey-biddle-pedo because of its simplicity, maybe in early phase trials that have less of a budget for lots of staff and fancy statisticians. But in the end, there's software now that just about anybody can do these more technical boundaries. You just need to do the reading so you understand how to do them correctly. Okay, so we've talked a bit about reasons to stop. You achieve your goals, you get convincing evidence early, you have safety signals. Another area that DSMB monitors, often at the request of the sponsor, is if it's futile to continue in the sense that the trend is overwhelmingly negative, no harm, no benefit. You've got 4,000 people lined up in the randomization. Are you going to wait how many years are you going to wait for a null result? If you can tell partway through that the data is so incredibly null that even if you accrue the next 500 patients, it can't reverse the null. You will never get below that stopping boundary or that significance at the end. That saves expense. It saves patients for more promising treatments in future trials. It frees up investigator time to go after those new other developments that would be more beneficial. So there's many reasons why you would actually want to think about what's called a conditional power analysis, which is given the trend so far, what's the likelihood you would have a positive result at the end given the planned analysis. And so that's one way of being futile. Other reasons might be you can just look at the recruitment rate and the writings on the wall. This grant ends in a year. Look, you're not getting 200 people from this rare disease. Event rates might be too low. There could be unexpected dropout or crossover. Sometimes you're studying something that's easily available, like screening. And so you might realize that you just can't achieve the scientific goal. The one area that's different than the rest, or what I've listed here, is poorer than expected treatment effect on the primary endpoint. So that's slightly different, or at least I'm trying to phrase it so it's apparently different. If you want to prove that something is poorer than expected, poorer than you would hope for, then that's an unconditional power. Like if you power the trial to have 90% power to detect at least a 25% reduction, if you fully enroll and don't achieve that, then you have you sort of 90% confidence in the sense that the treatment effect is lower than what you would power to detect. And that might be useful. For instance, a lot of people take glucosamine because they think it helps their joint pain, or they take St. John's wort because it helps, and they think it helps depression, or other treatments that might not be so innocuous, and you might want to convince the community to stop doing that these treatments might have, certainly expense, potentially harms, and false hopes. And so you might want to finish a trial even though you know it's going to be null, because you want that convincing null result, a meaningful null result, that you had the statistical power to have detected something greater than 25% reduction, and you didn't see it, so you know that with a good deal of certainty it's not there. That's an actual argument to continue the trial, as long as there's no safety compelling you to stop early. So the fact that you might be heading towards a null, that might be useful and not necessarily a reason to stop. It really depends on the circumstances. Typically, if that drug isn't on the market yet, and then it has no chance of approval unless you hit a target of efficacy, then you do futility monitoring for the purposes of stopping early and dropping that treatment. But if it's already in use and you want to convince the medical community, then I think that's sort of more on the scale of you wouldn't stop a trial for futility, or you have secondary endpoints that are just as important. A lot of times a trial has multiple goals, and as long as there's not a safety problem, you might not even monitor for futility of the primary if you know you're going to achieve something useful out of other endpoints. Okay, I think I've already covered that slide. So, this futility monitoring can be summarized by, you might want to ask two different possible questions. Will in the end the result be null? Will in the end the result be null and still meaningful? The first one is when you want to stop early. If you just, you might do a conditional power analysis. What's the probability of getting a null result, given the data that we have so far? And the other one, if you want a meaningful, interpretable null result that gives you information that the efficacy is lower, then you need the fully enroll, and you would rely on that unconditional power. Okay, so summarizing, monitoring recommendations. Once you look at the totality of evidence, efficacy, futility, safety, study validity, the board would think about making recommendations to continue the protocol unmodified, to continue but modify, potentially to terminate, and they summarize those and say a set of bullet points that they give to the investigators and the sponsor. The sponsor, in the end, makes the final decisions to adopt any of those recommendations, and certainly whether or not to terminate, as we've talked to before. Here at NIH, it's the institute director. that makes the final decision. And in general, we'll follow the recommendations of the DSMB. There are interesting examples in the Women's Health Initiative and the hormone replacement trials is a fascinating one to look into. I think they disbanded the first DSMB because they didn't like the recommendations. They wanted an independent second opinion. And you can imagine how that first DSMB felt. And so I would say this is, I mean, if you like suspense novels, I would reach for a case study of DSMBs because they can be cliffhangers and with lots of twists and turns. So ultimately, a DSMB could recommend stopping, which is the hugest possible outcome of a meeting. And that could be if the safety issue emerged, if efficacy was achieved, that's best case scenario. Interim results preclude a positive result, that's the fertility. This is a disappointing one, that operational difficulties are insurmountable, generally because either the primary endpoint was just infeasible and could not be obtained in enough people without and too much missing data, or accrual was just in the pits, and you're just not going to be able to add enough sites to finish on time. Interestingly, sometimes when there's multiple countries, multiple groups could be studying similar therapies. There could be other trials that provide information that give you information about what you're studying, either is no longer being needed or potentially sometimes you can get safety signals from other trials that were slightly ahead of you. And so that's important when you're on DSMBs to be aware of people who are other countries or other groups that are studying similar treatments. And oftentimes those DSMBs are formally put in contact with each other. And I think, hopefully, as we've, it's just this lecture always seems to fly by. There's so many issues to talk about, and I always feel like I didn't get them all in that I wanted to. But hopefully, you can appreciate, at least from the brief examples we've talked about, that the decision process of a DSMB is very complex. You have to worry about internal consistency, that this data is meeting expectations, or is consistent with the pilots going in. You think about external consistency. There might be other groups with related results. You're thinking about benefit and risk, the balance of the patients in your trial, who are ultimately only a small number compared to the total number of patients that they're representing, and how well do they represent those people in terms of their covariates being what you had hoped as being a representative group. And you think about that clinical and public health impact of having convincing evidence, not only that this treatment works, but to make people change their practice. And that can be quite an amount of additional evidence. And then, of course, the statistical issues of sorting out signal from the noise can be quite complex. And especially when you have rare side effects that are extremely worrisome, you don't want to wait until you have statistical evidence to prove without a doubt that you're harming people. And yet you also don't want to be swayed by one or two unusual events that might have been due to chance. And there's just no hard and fast rules to be able to sort that out. So it's a lot of hand-wringing. So I hope that really gets you interested in joining one of these DSMBs. They're incredibly important. They're voluntary efforts, and really it's about protecting the patients in that trial as a first responsibility. And the one thing that is also wanted to emphasize in terms of the complex decision-making of this body is you have that individual versus collective ethics, which perhaps you've already started that discussion in this class and will continue as you talk about IRBs, I think. That as you decide whether or not the patients are being best served, you're thinking both about, primarily you should be thinking about the people that are currently taking this drug and being exposed to risks, but you're also thinking about their future and the future of other patients as you make your decisions. And that's somehow having to work in that collective ethics. And on top of all the technical complexities of data and noise and signal, you have what can be complex cultural and ethical decisions to sort through, and something that recommending stopping should not be taken lightly for many reasons. And one of them is actually that stopping early handicaps safety analyses. Right, you're not getting the full amount of data that you could have had about this drug. It can actually affect it. They have shown statistically, essentially there's a little bit of bias in your treatment effect if you're stopping early for benefit as well. And people have fixes for that, but that's a little bit more esoteric. I think that the more complex one is that clinical trials are very good at comparing the arms of the people in the trial, and they're less good about making more absolute statements. And so sometimes a trial can end, such as the Women's Health Initiative, the hormone replacement trial. We learned women who basically started hormone therapy perhaps some years after going into menopause had excess stroke and excess, certainly no benefit in preventing breast cancer. And yet that raised, and of course, women immediately stopped taking these drugs. But more questions have arisen about, was that the right population to study? And many women would have started those drugs much earlier. Are the risks the same? And so sometimes stopping a trial is not necessarily the end of a question. And I did want to just finish with one last example before getting into some of the sort of nuts and bolts of the nitty-gritty of the meetings. And this has to do with that idea of, it's not just a statistical decision. The stopping boundaries, and a perfect example of that is well, I would have said recent, but I guess it's already going on 10 years. But this was a collection of trials that were done in Africa in terms of looking at whether or not male circumcision would prevent HIV acquisition. And so there were phase 3 trials, large trials, that began at the same time in South Africa, Kenya, and Uganda. Typically what they're doing is they're taking a group of people that they're recommending, and this is not a typical practice. And so there's a group of people that would be randomized to get usual care, and another group of people that are going to be randomized to be heavily counseled to take on circumcision, and you're randomized to basically say you would accept either outcome, so that you don't have a bunch of people that sort of got the wrong arm and quit. But in any case, they had several trials that they had going on in Africa, South Africa, Kenya, and Uganda. The designs were very similar, and this is a cultural practice, and so there was thought you would need to study it in multiple cultural settings in order to get an idea that this was going to be working in each of those settings. So the South African trial had a head start, and early on they had clear evidence of efficacy, and they stopped early. But I think the DSMB was quite sensitive to the fact that folks in Uganda may not accept a result about South Africans or think that it pertained to them, and there could be real cultural and behavioral reasons why it might not pertain to them. And so for many of those reasons, the DSMB decided to continue, even though they had evidence that in theory this would work in any individual in Africa. And what they wound up doing is increasing the frequency of monitoring the other countries so that they could stop as early as possible if that evidence occurred, and in fact it did in each of the other countries.